This is a fantastic turnout. Thank you all very much for coming along. This is our biggest one yet. So I'd like to say hello and welcome you here to today's seminars. Today's the fourth program update of an exciting BHP and Curtin University initiative that centres on the field of environmental DNA, or eDNA, and its use as a biomonitoring tool in biodiversity. I'm Shane Herbert. I'm the program manager for the initiative that the team are all going to be speaking on today, and I'll be the MC. But before we get started, I'd just like to make an acknowledgement. So Curtin University acknowledges all First Nations of this place we call Australia and recognises the many nations that have looked after the country for more than 60,000 years. We're honoured and grateful for the privilege to maintain campuses operating in Boorloo, Perth and Karkula in Kalgoorlie in Australia. We pay our respects to elders past, present, and as custodians of owners of these lands. We recognise their deep knowledge and their cultural, spiritual and educational practices and aspire to learn and teach in partnership with them. Curtin also acknowledges First Nations peoples connected with our global campuses. We're committed to working in partnership with all custodians and owners to strengthen and embed First Nation voices and perspectives in our decision making now and into the future. So I'd just like to address a few housekeeping matters. In the event of an emergency, please exit through the doors that you entered. There are stairs. Apologies. It's just the unmute. No one noticed. All right. Okay. Good. All right. Matthias just said it's all good. Great. In event of emergency, please exit through the doors that you entered, down the staircase, only down one level, down to the ground level, which is St George's Terrace. There's obviously an elevator there, but in the case of a fire, don't use the elevator, please. The muster point is south through Brookfield Place, across the footbridge, so hopefully you've got comfy shoes on, over Mounts Bay Road and over to the grass area near the Perth Convention and Exhibition Centre, so down here. Toilets are located through the door in the hallway, just off to my left. And I could also ask that you take a moment to check your mobile phones are set to silent. We've organised refreshments at the conclusions of the presentations. However, in the meantime, there's still and sparkling water at the back of the room. I don't remember checking, I think that's there. And there's also dispenser taps in the kitchen just off the hallway as well. So please help yourselves using the glasses that are available. Importantly, refreshments are downstairs at W. Churchill's from 4 p.m. on, and I'd encourage you all to please stay and network with everybody. It's been a great group of people and we have some great conversations afterwards. So that's down on the basement here, and we'll have some of the team directing you how to find that one. This presentation is also being broadcast by WebEx. We've muted the lines, but if there are any questions for the speakers, if you could put them into the chat, and my colleague Sam here, we'll pick them up and put them to the speaker when we have any. We also aim to capture today's proceedings in a recording and make it available afterwards. If anyone has any privacy concerns, then please let myself know or some of the Curtin team, and I'll do our best to cater for that. Okay, thank you for your understanding. I think with now all that said, we can begin the presentations. So today's lineup, we've got seven presentations across the different projects. First up, we're going to go in order of the projects that we've named them, 1 through to 7. Pilbara Olive Python will be Pilbara Olive Python detection and conservation, subterranean fauna, then the Hypersaline lakes, focusing on the Chile Hypersaline lakes, improving sensitivity and specificity of eDNA tools for invasive species detection, and then its application on benthic communities and infrastructure in the Pilbara. Then terrestrial ecosystem biomonitoring with eDNA across the Tree of Life, focusing on the Olympic Dam study site. And then a new project being introduced, which is called Living Waters Western Australia, or LIWA. So, what I might do now is hand over to Professor Prof Allentoft to the stand. Morton's the leader of the Trace and Environmental DNA Lab, in which this program is housed at Curtin University. Morton will be doing a brief introduction to the program's aspirations and outcomes before the teams delve deeper into the studies. So, over to you, Morton.
Thanks very much, Shane. And thanks everybody for showing up. This is a great turnout, actually. I can't believe that we're standing here now again. 4 years it's been, right? 4 years? It has been 4 years. And I'm telling you, the first 2 years was a pretty bumpy ride for all of us because we pretty much signed the contract just before COVID hit the world. And I was sitting in Denmark, just hired at Curtin, and had to try and manage this lab from a couch in Northern Europe for the first year and a half. Somehow we came through, and all the research we'll be presenting here today is kind of concludes the eDGES project. As Shane has already alluded to, a couple of the projects are still ongoing, so it's not entirely done by 4 p.m. today, but we're getting there. Okay. Trend Lab at Curtin University. Most of you are, of course, familiar with that. We even have the founder in the room today. Mike Bunce is sitting here somewhere. And about 4 years ago, I took over from Mike and has tortured his lab ever since. It's a beautiful place to work. We have a good batch of people, around 20-25 people doing excellent research in Trend Lab, I would like to believe. And we have very good molecular resources available at Curtin University. eDGES is hosted in Trend Lab. It's a tight collaboration between Trace and Environmental DNA Lab and then eDNA Frontiers, where Shane is the business manager. And then I would also highlight, of course, Euan Harvey, who's co-leading the eDGES program together with me. Euan, I couldn't find the fish ecology logo to put on here, but I'm sure there would be one somewhere. All right. Just to briefly highlight what we do in Trend Lab, eDNA, environmental DNA, that's why we're here today. And this is, of course, a very big and heavy component of the work we do in Trend. We do, however, dabble in other aspects of molecular ecology, I would say. We do some ancient DNA research to ask what's in the past, eDNA to look at the present, and then we do dabble a little bit with genomics also to look at how the organisms have actually evolved to cope with whatever environment they are living in today, right? And those three aspects together is kind of making up a holistic way of looking at molecular ecology. This is where we are aspiring to go. And you will see eDGES today. It's very eDNA focused, but some of the projects are kind of bridging the gap between these different disciplines, right? So we are making a move in that aspect. eDNA, I think we have shown this slide countless of times, so I'm honestly not going to stand here and explain to you in great detail what environmental DNA is again. But just very briefly, you know, the concept when an organism dies or defecates, urinates, shed fur or whatever, they will leave old cells in the environment. Inside these cells, you would have DNA, and each organism has a unique DNA profile, right? So if you pick out those particular regions of the DNA that is unique to the organisms, you can sequence them, and you can identify which species are present. And that is, in a nutshell, what it's about. It's about using environmental DNA from all kinds of substrates and environments to identify which species are present. Then it becomes a biomonitoring tool for looking at biodiversity shifts or understanding what species are present in space and time in whatever environment you're surveying. So, in regard to the eDGES, I think this is roughly how we phrased it in the initial project description. You know, what is eDGES really about? What is our aims? What do we want to go with this? And I'm just briefly going to read these aloud. I'm not going to elaborate too much because you will see all the individual projects doing exactly these things anyway in a second. So eDGES is about developing and applying environmental DNA techniques to address biodiversity loss and sustainability. We seek to understand ecological and evolutionary changes in natural and man-made landscapes. And this is where I would say the molecular disciplines are being bridged a little bit, because it's not only about looking at species presence absent, but actually also understanding evolutionary relationships, sequencing genomes, going back in time to try and understand how these organisms are adapted, and potentially what also makes them go extinct. We want to develop new generation tools for managing species, threatened species, and we want to do biodiversity conservation through whole community studies. Some of these projects are very species focused on single species. Some of them are doing broad scale inventorying of the biodiversity in a given region. And then, of course, as you all know or should know, this is supported by BHP's social investment framework, and that's why we're here today, basically. It's projects across 4 different environments. We have a couple of terrestrial projects, as you will see. We have dabbling with freshwater eDNA, we even go in underground and look at subterranean fauna, and then we have 2 marine projects also that will be presented. I'm not going to say more about that because you will hear everything from the researchers, the other researchers in a second. So we started with 5 projects in eDGES. This has then expanded a little bit over time. So we actually have 7 projects now. And as I mentioned, not all of them will end today. Some of them will, but a couple of projects are actually ongoing. And these are projects that each builds and expands on eDNA's utility as a monitoring tool. In terms of the outcomes, I would say that we have listed 4 key outcomes up here. It's always high impact science and peer-reviewed papers. That's we are researchers, right? We're running a research lab, and that's kind of the core of everything we do. It's not the only thing that's important, but if you cannot deliver high quality research, then everything else that we do will kind of fall apart. We cannot do community education and awareness. We cannot do input to policy if we don't have that foundation in place, right? So any project we'll be doing in Trend Lab will have that focus on being able to deliver high quality foundational research. And then I would say some of these other aspects, we believe that we have delivered fairly well, I would say, as you will also see in this in that first category there. Some of the other ones about community education and awareness, input to policy, are perhaps a little bit underperformed, I would say, in this eDGES project. And it's not actually that surprising. This has been a big, complex project. It's taken a while to actually learn how to manage that. It's taken a while to learn in this framework to produce high quality science. And now, as you will see today, we are actually ready to move into some of these next categories with that research foundation in place, I would say. So this is moving rightly in that direction. And then I just want to say, and I may be jumping the gun a little bit because Tim might say at the end of the day that this has all been a waste of money. I don't hope he will do that. But I do believe, and this is the general vibe we're getting, at least from him, that it's actually worked quite well. So I hope you will agree, Tim. And I just reflected a little bit last night about why has it actually worked. As I mentioned, it's been quite a bumpy road for us with COVID and all kinds of other things, challenging collaborations. But I do think there are 4 key aspects here that has really been in place that has really worked really well for the eDGES project. First of all, people. You don't get anywhere without people, right? That's in any kind of connection, any successful project. We need to have people. And we have been so fortunate and lucky in the eDGES program to have some very skilled people working with us in Trend Lab and elsewhere. And not only skilled, but also very good human beings that are wonderful to work with. And this has been an amazing journey for all of us, I know. And one of the reasons why I think, key reasons why we have done quite well. And then trust and freedom. I think this is freedom. Let me start with that one. This is quite important, I think, for all kinds of people. We don't like to be micromanaged. Perhaps this is even more important for academics. I don't know. But I would say the way that the collaboration with Tim and his team at BHP has worked have really been tremendously based on freedom and trust, right? You can probably imagine when you're a bunch of conservation biologists working together with a mining company, things can go wrong if you don't have a strong trust bond between you. And we've had that from day one, I would say. It's been incredible to actually see that amount of trust that Tim has given us and the freedom to explore and go in those directions that we have judged was important in order to deliver the outcomes that we had promised, right? So we've had a lot of freedom to navigate in that space. And then, of course, resources, that goes without saying. You don't get anywhere without the money, you don't get anywhere without the labs. And we've been so fortunate that BHP and Curtin University have supported us generously over these last 4 years. So I would say that this has been in place, and it's been a real privilege to operate in that intersphere between these different important components here. Okay, that was it. I thought I had another slide, but I don't. Thank you so much, everybody. I think I will just hand over the word. So will you do that, Shane? Brilliant. Okay, so let's. Thanks, guys. This was and welcome once again. I will give the word to Ben and Marsa, who will talk about pythons, my favorite creatures.
Hello, everyone. Good afternoon, and thank you for joining us today. So on behalf of our entire team on eDGES Project 1, which is on Pilbara Olive Python, biomonitoring, and conservation genomics, Ben and I are going to present our progress to date on this project. Thank you. And also, as Morten mentioned, this is one of those projects that is not going to end today because that's part of Ben's PhD project, and it's going to go on for at least another year and a half. So with that, let's just start. As you may know, olive python is one of the largest snakes in the world, and they're also a top predator in their habitat. In Australia, we have two subspecies, one which is commonly known as Kimberley subspecies, has a wider distribution, and then we have the Pilbara subspecies, which is confined to the Pilbara region, and unfortunately, it is listed as vulnerable. And that's due to various factors, but perhaps one of the most important reasons is the loss of habitat because there are continuous development in the region and also land use change. But on top of that, these species, or in general, snakes, they are not really easy to find in the wild and to study in the wild, which is why our project aimed to first improve the biomonitoring for these species, to say, okay, can we actually detect them using eDNA methods or not? Also to develop high-precision biomonitoring tools, that being a qPCR assay, so we can be more precise in terms of presence, absence of these species. And also, we wanted to develop an eGenomics framework that can bridge the gap between eDNA and genomic science. And Ben and I will just take you through those steps shortly. With respect to our first aim, which was, can we actually detect these pythons from eDNA samples, which we can take from rock pool waters, we were quite lucky, and thanks to few consultation companies and their clients, including BHP, we were able to actually collate some eDNA samples that these companies had collected over a few years as part of a wider biomonitoring program in the Pilbara region. And then we were able to just run through our own metabarcoding pipeline on those water samples and take it all the way through. With that, we could discover a variety of taxa, including our beloved olive pythons. But I don't want to really go through the details of that project because we presented all of these results last year, and on top of that, it is actually published, so if you are interested and need a refresher, you can just check our paper again. But then, now that we know, okay, we can actually detect them using these water samples in the rock pool, we wanted to check, are there any other substrates that may act as a good alternative or they could complement what we found using water samples? Can we actually improve our detection rate if we use some other substrates? And for that, we needed some systematic testing.
So to do so, we went to Perth Zoo and we tested the olive python enclosure and the Burmese python enclosure as an outgroup. That's a lovely picture of us there. And we collected spiderwebs, soil, filtered air, and swabbed rock and tree surfaces. And I'm currently metabarcoding those samples, so we will learn how they perform as opposed to water and what kind of biodiversity they capture and sort of give us the full picture. Of course, we also want to narrow in on the target species. So we have also developed a qPCR assay for the Pilbara Olive Python per our project aims. And this is a highly specific, very sensitive test where you can very quickly get results, like in the bottom left. And it's just, is it there, is it not there? And we also tested this on the zoo olive python enclosure. That's the plot in the right. And here each substrate is along the bottom. And then we've got, on the y-axis, the lower it is, the better. It's a measure of how quickly it starts to amplify. So as you can see, we weren't able to get water from this one, but we did get water from other enclosures. And we can see that spiderwebs performed excellently. Actually, it's quite surprising, along with surface swabbing from rocks and trees. And this already gives us an indication of which direction we should be heading and a kind of practical application if you can't reach a sort of crevice in a rock, swab it, maybe get a spiderweb if you can see it. It's really quite cool. But these methods, both metabarcoding and qPCR, both focus on just one very short region.
So as Ben mentioned, what's currently happening in most biomonitoring studies is that we are really reliant on a very small portion of often mitochondrial genome, which is usually, depending on what type of marker you are using, it is less than 300 base pair. But for a snake such as our Pilbara Olive Python, which has a whole genome size of 1.5 gigabase pair, that means that we are only using a very, very small portion of the genomic information that is available, because we know that in an environmental sample, it is not only those barcodes that are floating around, but we have the whole genome information. But to be able to pull those information out of our environmental DNA samples, we need a reference genome. So in collaboration with PERSU, which provided a high-quality blood sample, and also Vertebrate Genome Laboratory in the US, we were able to sequence and assemble a very high-quality reference genome for the first time for the Pilbara Olive Python. And what you really need to know about this table is just we have a very high-quality reference genome. So it's been assembled to chromosome level, to 17 chromosomes, and for those of you who are interested in numbers, this scaffold N50 number means that essentially half of the genome is assembled and is made up of fragments that are above 203 megabase pair. So that is just to say this is a very high quality genome. And with that, now that we have such a high quality genome, we can go ahead and test more novel ideas.
So, as some of you may recall, I did my honors on the decay rate of Pilbara Olive Python DNA in two environments, the pure and lake water tanks. And back then we got a very sharp drop-off in the lake tank. But this data was generated using qPCR, so one of the small regions just mentioned. And we selected, what we've done is we selected samples over this month and shotgun sequenced them to generate data over the whole genome. And we've mapped that shotgun data against the entire Pilbara Olive Python reference genome to basically get a proportion of the total reads in the sample that are mapping to the genome. On the left is the pure tank, on the right is the lake tank. And what we can see is that we get a really sharp drop-off, a very steep decline, but from 10 times less to start with in the lake tank, just due to the microbial decay and other DNA binding. Of course, this doesn't sound like a lot, but it's millions of reads regardless. We've got so much information, it's hard to fathom. But it's a very zoomed out perspective. So there's a lot going on here, I know, but we took the same samples and we mapped them to the Pilbara Olive Python genome, along with 4 other representative vertebrate genomes, to see how they perform basically in relative to each other. This is just proof of concept, but here we have the pure tank on the top and the lake tank on the bottom. And then in each sample, we have mapped all the sequences, all the DNA, to those genomes. So we see a very strong signal within a day, and then it starts to drop away as you get this random mapping as the DNA decays. It sort of starts not really caring what it's mapping to. Smaller. And I am about to use this on the zoo samples as well. So again, it's very preliminary at this point, but it's enough for us to say, good, this sort of works. We can actually test it in a real environment. Of course, this is just one aspect of the genome. Another thing we're going to do is population genomics. This is really interesting.
So having a very high quality reference genome means that now we can actually do larger scale study. We can go and sample a lot of individuals from between and within populations, and we can study various genetics concepts on those, which is going to help us to refine our conservation and management strategies.
So to sum up, basically, this is where we're at right now. As Master said, I've still got 1.5 to 2 years in my PhD, but I think we're really going well. We've got successful metabarcoded rock pools. We've proven that we can actually detect them in the wild. We're working on the substrate testing. We've got this high precision qPCR assay. So it's really specific, really sensitive. We can say, yes, it's here. And we are developing this eGenomics framework as well to sort of go further than just that one region. We've generated that genome. We're shotgun sequencing eDNA samples. And within that, we are using population genomics to see what has actually happened between then and now. Thank you.
Thank you very much, Ben and Marsa. Was there any questions for Ben and Marsa? Kat?
Beat you to it, sorry. Yeah, I'm just, great talk. Just curious about the size of the fragments that you are mapping back to your reference genome from the eDNA. And secondarily, in regards to the genomic application, what happens if you have multiple individuals in the same sample, the same pool, rock pool, essentially?
So in terms of the lengths of fragments, because this is just our preliminary studies, we haven't looked into the actual fragment comparison yet. But in terms of how we are going to filter out all those other individuals, so that will be sort of a repetitive mapping plan that we have. So obviously the first step is that we want to map and pool every sequences that belong to our Python reference genome. And once we do that, then we need to be sure that it's just not some random region. So we again take it through repetitive mapping to some other genomes, for instance, human, because that's like a big contamination, and also some other species as well. And we just try to refine the number of reads that are being mapped. So we can be pretty much sure that what we are collecting is definitely relevant to our Python genome.
So by the fragment length, did you mean the qPCR, the metabarcoding?
The shotgun sequences, yeah.
So wasn't sure.
Yeah, great talk. Thank you. In terms of the sampling methods, I'm just wondering how you think the methods used for spider webs and rock surfaces and tree surfaces will translate to the field in the Pilbara. Like the spider webs that you were using, are they from the burrowing spiders, and are they present in the Pilbara? And the rocks and the trees, were they wet or very dry? And do you think that'll also translate to the field? So just interested to know what your thoughts are.
So the spider webs and the rock, or the rollers specifically, they have both had really good papers really recently. The spider webs in the zoo enclosures were just in the air. As you may have noticed, they performed way better than actual air filtering, but they're basically just like an air filter that's been around for a much, much longer period and stickier, I guess. The rock and tree rollers, you dampen them a bit beforehand just to help pick it up a bit better. But other than that, it was just rock and tree surfaces from all areas of the tank.
Okay. All right. Thank you. Well, if any other questions, then these guys will be around at the end for more questions. Thank you. So next up, I'd like to introduce Dr. Nicole White. She'll be talking on eDNA for subterranean fauna detection and conservation. Did you want that?
Hi, good afternoon. Thank you, everyone. So we've heard a terrestrial project. Now I'm going subterranean with you all today. So this is where I've spent most of my time over the last 3 years. and I had the opportunity and have built an excellent collaborative team who all have similar interests in subterranean fauna, their conservation, detection, taxonomy, describing species. And it's been great. It's actually been really, really great. So today, I wanted to showcase basically all the work that's being done under the Subterranean research umbrella. So I've been working with Michelle Guzik from the University of Adelaide and Mika on an ARC linkage grant. Both the eDGES program and the linkage grant have been running side by side. And then we've had a few students, and then of course, Julia Perina, who is a specialist in her area with taxonomy and describing species, and then a little pilot project that Jason and I got into a little bit as of recent. So the Great Artesian Basin, when I started this project, BHP said, could you actually start in South Australia first? COVID. So that made things a little bit challenging because I couldn't actually travel over to South Australia. Michelle and the BHP team were over there, so I was fortunate enough that they were able to go out and collect the samples. So it's quite cool, and the main thing that I wanted to show here is what I was really interested in is if these springs, for some reason, start losing their biodiversity in the subterranean or the spring fauna, will they be able to be replenished from the deep depths of water underground? And as the graph over there shows, we have most of the genetic diversity that's in the spring surface. So these are actually very, very unique, and they're almost like little evolutionary islands. So the springs have been around a long time, and the flora and the fauna have evolved with the spring. So they're quite cool. And as you can see, the Great Artesian Basin is a massive area of Australia. It's a huge water resource. And we were interested in how do we protect the spring fauna. So this is what this graph said. So I looked at water from the deep and then also the surface water there. And there is a clear difference. The metabarcoding work, we have quite a large data set now that has 3 metabarcoding assays, the 18S, CO1, and bacterial 16S, and we're still doing our analysis on that data set. So really my aim for the project was to start generating reference barcodes, but not your typical 600 base pair CO1 barcode. All right, so what I tried to develop and seem to have gotten going in the lab is shotgun sequencing of subterranean fauna so that we could generate the mitochondrial genomes. And I'm always surprised when I get the data back and start looking at these mitochondrial genomes because I find surprises. Just as you can see on these 2 screens, we have 2 genes, which are ribosomal RNA that are in the red, and you can just see between these 2 animals, an ostracod and an isopod, that the gene order, they've had major gene rearrangements. And why do I say this? This comes very important when you're trying to develop eDNA assays, markers that are targeting that barcode gene, or when we're doing eDNA work, the short regions. If you've had major gene rearrangements, not your standard primers are going to work in the assay. So this is actually giving us another insight into how we can start not only getting a whole mitochondrial genome for a reference, but how we can start optimizing our assays a little bit more. So these are two creatures from the Great Artesian Basin. I've also been starting work on Ethel Gorge, and these are two animals from Ethel Gorge. And again, I look at these two animals and their gene rearrangements are different. So what do we have now over the last almost 4 years? We have about 170 complete mitochondrial genomes, so that's quite a wealth of resources, and it's still ongoing. I have more work to do. So it's a bit of a stay tuned, but we are developing now a mitochondrial reference database for Stygofauna. So I wanted to showcase a little bit of work that Milad has been doing. He's enrolled through the University of Adelaide under the ARC linkage grant. I'm an external supervisor for Milad. And he became quite interested in gudgeon. He had a bit of a fish DNA background, and he's been working on developing the gudgeon assay. So I generated the mitochondrial genomes. I got some tissue from the WA Museum. I shared those with Milad so that he could start developing a gudgeon assay for detection, because it's not only invertebrates that are underground. We have some vertebrates that live underground and in the dark. And as you can see, this fish over there has no eyes, because when you live in the dark, you don't need to see. So they have lost their eyes. They may have had them at one time, but they have lost their eyes. So from what we can see here, we have a pretty well-validated gudgeon assay, and Milad is just putting the paper together for his research program. Did Michelle's picture not come up? Oh, sorry, Michelle, if you're online. Michelle is in transit. She's coming over to Perth. She arrives tonight. Her apologies that she couldn't be with us today. This is some work that Michelle has been working on. So what we were finding and what Michelle had identified early is a lot of the consultants, they'll go up and they'll do some work, they generate some barcodes, the standard barcode, and then where is the data? We rely on these reference barcodes when we're doing environmental DNA work, because we sequence everything underground, but we don't know what it is that we sequence. We need to compare it to a database that has high fidelity, so we can be pretty confident with our answers when we're matching the DNA sequences. So Michelle has been spending a lot of time working with BOLTS, which is the barcode of life. We now have 5,910 curated CO1 sequences for subterranean fauna, and it's growing. And I hope to also incorporate all the mitochondrial genomes into the BOLT database. So this is where Michelle's been spending a lot of her time, also running the ARC linkage program, which was quite a large program. There she is. Mika. So we had this 5,910 reference CO1 barcodes. Mika picked up all that data and ran a program and found all of these already previously published assays that either target arthropods, they were called universal, or invertebrate. And this program looks at the sequences from all of the orders or groups that are across the top, and then matches how well that assay, the primer, will match that sequence and actually amplify that group. So when you see green, that's good. The primer should work and amplify that group of subterranean fauna. Where you see red, it's not going to bind. Okay, so then Mika went through and she validated and tested 31 primer sets. And we landed on the best primer set. So even though up in there, a couple of primer sets were identified as being really good, But when we actually put them to the test, so this is just binding, but not necessarily sensitivity. So when we put them to the test, there was another primer set that looked really good, but it needed quite a high copy number of the target DNA to be in the sample to amplify. All right? So then Mika tested another one, and it seems that the FWH-F2 and FWH-R2N seem to be quite a good, doesn't get all of the groups, but does do quite a broad sweep so that we can grab the subterranean fauna and start characterizing these underground systems. Okay, then we are over to Jake. Jake did honors. He's enrolled through the University of Adelaide and has just started his PhD, and he's staying in subterranean and underground waters. But what I wanted to show here is when we shotgun sequence subterranean animals, because they're so small, I have no way of treating the exterior of the animal. So I'm sequencing a lot of bacteria, whatever the animal has on its body from the environment. I'm also sequencing everything that that animal has in its gut content. So it's a shotgun sequence. So not only that, it just, when you shotgun sequence, it doesn't just sequence the mitochondrial DNA, we're sequencing nuclear DNA. And what Jake did is he went in and started mining. After I pulled out the mitochondrial genomes for what I wanted, he went in and mined the 18S. So you can also go in and start pulling out more of the data for other reference genes. And then he just put together this nice 18S sequence tree, and this is mainly focusing on subterranean fauna in Barrow Island. Julia is doing her ABRS grant through Curtin University through the Surge Subterranean team here. She is an expert and has the expert eye. She is describing parabathanelids. She can tell you that they all look the same, so she spends countless hours pulling these little guys apart. And as you can see, the red circle there, that is an animal on the microscope slide there, that little dot, and she will pull that apart in 20 pieces and make her slides to do her taxonomic drawings. So Julia is very, very busy in this group of animals. And the last one is a little air eDNA sampling for troglofauna. We cannot forget the troglofauna. I've been focusing on basically stygofauna. They're aquatic, but we also have this troglofauna that lives in the cracks and they're air breathing. Jason came up with this idea. He engineered it. And we did a little trial run. Can we detect troglofauna species through this fancy air filter? Then that's then dropped down through a bore that has been drilled. And preliminary studies is showing good promise with this project. So with that, and that very quick showcase, I can say that I'm very happy. We've substantially grown the eDNA toolkit for subfauna. There's a lot more work that needs to be done, but we're coming at it multi-pronged, and we have a great team who has a great enthusiasm for everything that's going on underneath our feet. That's very important. Basically, we have 9 publications and more to come. So the barcode reference library, the CO1 barcoding assay, all of that is up there if you're interested. And then a big thank you from the Subterranean team. And I'll open it up for questions.
Thank you, Nicole. All right, we might have time for one quick question if anyone has one. Ollie?
Hi, Ollie.
Thank you. It's reference
Genomes.
genome question.
Yes.
And that photo of that microorganism made me wonder, is there scope for vouchers?
On the slide?
Yeah, like,
Yes.
so connecting taxonomy with reference sequences is even more powerful if you can attach it to vouchered specimens. So I'm just wondering
Absolutely.
how you guys are managing that.
That's what we're
be a
trying,
challenge.
yes, that's what we're trying to do. So when the subterranean fauna is collected, I'm trying to get the identification down as far as I can. I can't do that, though, myself, so we're relying on the experts. And at the same time, for that to happen, we also got to maintain DNA preservation. So we're still trying to work that out. Like, I got a lot of animals from the Gab Spring that didn't go past the family level for the identification through a consultant. But yet, from Ethel Gorge, I'm getting animals to genus and species for mitogenome sequencing. So. No, we have to get multiples of, and that's what I'm asking them for. So you're describing this one, but this one's identical. Can I have that one? Yeah. 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 It's a challenge.
Okay, thank you very much.
Thank you.
Cheers. Okay, the next talk is online and it's recorded. So Mattia's currently out of the States, so which is the case with a large group. And I was going to say, yeah, it starts playing as soon as we advance the slide. Mattia's online for questions afterwards.
Seminar. By the way, I'm sick, so I'm kind of glad that I'm not spreading my germs right now in the room. Project 3 focuses on the functional ecology of hypersaline lakes by means of multidisciplinary approaches. Just a bit of a background before I outline the main findings of the project. Hypersaline wetlands are definitely worth our salt. If we consider that saline lakes account for almost half of the volume of inland waters, and therefore salinity is one of the key biodiversity drivers all over the world. Saline and hypersaline lakes also host high-range biodiversity if we consider microbial communities especially, but also metazoans such as invertebrates or flamingos. Overall, they are constantly and increasingly subjected to impacts such as climate change and extractive processes, and this is actually affecting the long-term ecological stability of these ecosystems. Here we have a few examples on the extractive processes in terms of mineral exploration, for example, also artemia harvesting in the picture labeled as B, but also I wanted to highlight the ecosystem services provided by this unique environment, such as recreational and also ecotourism purposes. The project counts on the collaboration with the University of Antofagasta and also the partnership with Chilean regulators such as CONAF. There's a PhD student, Gonzalo Salazar, who's entirely funded in terms of salary and also analytical funds throughout the project, and he expects to finish the project actually in 2026. Now, the relationship between eDNA and hypersaline lakes is a challenging one. Hypersaline lakes, by definition, are polyextreme environments, thus high salinity. The definition by the book of hypersaline lakes involves concentration of salts beyond 35 grams per liter. The exposure, for example, in the Atacama region is the highest in the world, and there's also difficulties in filtering water when the salt concentration is high. Due to these characteristics and the need of establishing protocols, but also due to the difficulties associated with COVID times, we focused initially on Rottnest Island for a few pilot studies, and then we established the core research in the Chilean Precorded Journal. There's also another in parallel goal in terms of the analysis of food web interactions through the use of stable isotope analysis. Overall, two goals were involved. We wanted to test, validate, and assess the use of eDNA in hypersaline lakes, and also investigate functional ecology patterns through the use of multidisciplinary approaches. Let's have a look now briefly at the main findings of the Rottnest Island satellite study. We collected water samples from 5 hypersaline lakes located within the island, and we focused on bacterial and archaeal communities through the use of the universal primer Bacteria 16S. 3 were the main clusters grouping these 5 lakes, and 2 were the drivers shaping this clustering. Total dissolved solids, which is an indicator of salinity and also alkalinity. In terms of functional patterns, we went from moderately to extremely halophilic groups. And overall, what does this mean? Well, from a conservational management perspective, if we think about Rotterdam Island Authority, these results are indicating that we should focus on a lake-by-lake basis and not consider this just one giant salty pond. Specifically, Garden Lake was the one that was most affected by contamination and therefore requiring more urgent ecological restoration. The second stage of the Rotterdam Island Satellite Project involved a collection of macroinvertebrate samples through nets. And the combination of a metabarcoding approach, CO1 in this case, and a morphology-based approach, the conventional ones, described 16 taxa overall. And the main take-home message of these results was that there's great potential behind the combination of modern eDNA and conventional approaches. If we have a look at the table comparing the results from net versus metabarcoding and findings for the 5 lakes, some of the most remarkable findings were, for example, that sea monkeys were dominating all the systems, both from net and metabarcoding. With eDNA, we were able to find exotic species such as Aedes atraculus, or endangered species such as Haloniscus cerulei. This has great value in terms of the ability of eDNA in detecting certain taxa that are actually not usually detected through conventional approaches. Quickly moving to the Chilean part of Project 3, we focus on 3 hypersaline lakes located within the Flamencos National Reserve in Chile, Sala de Tana, Sala de Puxa, and Laguna Pular. We targeted both water and sediment, and samples were collected in 2021. Overall, this batch of samples provide the first multi-eDNA assay analysis in water and sediment for this region, and as far as we know, the eDNA samples from the specific salt lakes, Sala de Puxa, were the ones that were collected at the highest altitude in the literature. The microbial communities at the 3 hypersaline lakes were quite different. At Puxa, there was a dominance of just 2 phyla, while at Pular, we had the vastest proportion of unclassified bacteria. Stay, but there.
Your son is a very like sleeper, so he's within these global wish, but what's happening? He person the man record the circus.
All right, she's doing things. Things are happening.
Yeah. Well, a bit.
It's so funny watching someone do stuff on the computer.
Yes. Thanks for it.
Think about 4 minutes.
And at Tana, the proportion of archaea was considerably higher compared to the other 2 hypersaline lakes. Specifically, at Tana, we found a high level of heterogeneity in between the sampling points collected at the same lake, indicating that this lake in particular is providing a vast set of heterogeneous environmental conditions, potentially triggering higher degrees of diversity. Once we have a closer look at metazone diversity, eDNA analysis revealed a total of 21 and 22 taxa across 3 hypersaline lakes in sediment and water respectively. Tara is the most biodiverse in terms of metazone communities, with 15 and 13 taxa from sediment and water respectively. Now, our multi-assay design, in this case we use 12S for birds, 18S and CO1 for vertebrates and invertebrates, was able to detect a range of resident hypersaline taxa with different conservation status, spanning from rotifers, in the case of Excentrum, to endangered nates, as the case of Heliobia atacanamensis, to amphipods, all the way to flamingos, in the case of the genus Phenicopterus. Now, beyond all these graphs and patterns, 2 main take-home messages can be extracted of Project 3. First, hypersaline lakes are rich, and they're rich beyond minerals. In fact, they host a diverse set of microbial and invertebrate community that are actually providing the food sources for wading birds. The second take-home message is that high-salt eDNA is very insightful. This technique provides a powerful complementary platform for unveiling otherwise very much overlooked biodiversity and functional patterns. In terms of more tangible outcome of Project 3, there were 7 direct and indirect publications associated with the specific project. There were a few scientific oral expositions and posters, and this project also served as the ground for new, fruitful and interesting collaborations all over the world involving Russia, Italy, China or India. Metaphorically speaking, I consider that eDNA analysis is doing to nature conservation what the email did to communication. In fact, eDNA is cost-effective, is non-destructive, or at least minimally destructive, exploded in recent years, but is definitely established. It coexists with conventional technologies and is applied in many contexts. It can also be combined with other tools. Well, once we add a bit of salt in forms of saline and hypersaline lakes, I also consider that this metaphor gets even more powerful. And this is absolutely crucial given the current paradox of lithium. Lithium is an essential resource for a greener economy and a greener world at the end of the day. But the extraction of lithium comes at a cost, which is a very high cost for saline and hypersaline lakes, while eDNA is essential in bringing new light on the ecological functioning of saline and hypersaline lakes, and therefore can be really useful in framing targeted and really effective conservation strategies. Some future perspectives involve more data to come of Gonzalo's PhD on food interactions and also the diet of Brazilian flamingos, the analysis of the microbial and proteus diversity across the same salars, and now the potential of eDNA techniques in saline and hypersaline lakes is unlocked. Next steps could involve biodiversity monitoring over time or the design of biospecific assays, for example. Last but not least, I would like to thank all the collaborators involved in this multidisciplinary, at times challenging, but I consider very fruitful project, and looking forward for the discussion, networking, interaction, and questions.
Professor Simon Jarman, he's leading the improving in sensitivity and specificity of eDNA tools for invasive species detection. And then I believe Professor Euan Harvey will follow, talking benthic communities associated with infrastructure in the Pilbara. So you guys will tag team.
Fantastic. Thanks, Shane. Can you hear me okay, like that? Good. So yeah, this is a project about improving methods for detecting invasive marine species. It's done by a team of people, most of us at Curtin University, so me, Cheryl, Fred Wells and Euan are all Curtin researchers. Justin McDonald works for the state government here in Western Australia. So marine species that are invasive have been a particular problem for over the last 50 to 100 years. They were probably around before that, but we've been noticing them far more frequently. These two examples are real exemplars of this. So the one at the top, the North Pacific Sea Star, was first detected in Australia in the mid-80s around Port Phillip Bay and in Tasmania as well. It eats a whole bunch of things, but mostly the things it eats that we notice are habitat-forming organisms, things like the ascidians that the handfish uses to lay its eggs in. So this is one of the reasons that these handfish are going extinct around Tasmania, on the road to extinction. Eats kelps and things as well. The lionfish is originally from the Indian Ocean, but it's an invasive in both the Mediterranean and the Caribbean and Gulf of Mexico regions. It's thought to have been introduced by the aquarium trade and has become a real problem in those areas because it kills other fish. So it's completely disrupted fish communities in those areas, including many of the fish that maintain coral reefs, things that feed on the sponges and algae that don't get grazed down by them anymore, so that the coral gets outcompeted by other organisms in those areas. So very destructive organisms. A lot of you will have heard of these before, of course. How do I do it? Great, thanks. And of course, the reasons that things are invasive are getting worse. So this graph at the top is a graph of global GDP over time and the proportion of it that is related to trade. And since the time that those invasive organisms had become discovered around Australia and in Florida for the lionfish, it's doubled, basically. So trade accounts for a quarter of global GDP at the moment. That leads to a lot of shipping. So there are ships going everywhere, all around the world. Of course, planes can introduce things as well, but ships and all of those marine movable structures are a real problem for invasive marine species, of course. The other issue that we have, of course, is the oceans are warming. This causes a multitude of problems. So the primary thing that you would think of is that it moves things around. It changes the distribution of ocean currents. So this is the East Australian current here, showing that at one of the times in the summer when it pushes down towards Tasmania, it moves down that east coast at a regular time every year, but it keeps on pushing further and further south. This leads to lots of habitat destruction because all the kelps get killed and various other things that have temperature intolerances. That creates opportunities for invasive marine species to establish and introduces new species as the distribution of things that live in that warmer water change. So there are lots of problems associated with the changing world we live in, of course. So this graph shows an analysis of the number of families of different phyla of potential invasive marine species that are present in each phylum and thought to be potentially invasive in Western Australia. So this is an analysis that the state government did. So you can see at the top that annelids have, what's that, maybe 3 families of potential invasives that are thought to be a potential problem for Western Australia. Arthropods have a huge amount, chordates have a reasonable amount, and so on and so on. But outstanding from this graph, you can see that arthropods and mollusks have the biggest potential for invasiveness within Western Australia. And one of the big problems that we have, of course, is that for genetic data, we mostly have genetic data for chordates and things that are related to them. At least going back in time we did. There's a lot more coming through now. But one of the things that we need to do is come up with better eDNA tests for detecting things that aren't chordates and they're close relatives. So echinoderms like that North Pacific sea star are fairly closely related to chordates. So the eDNA tests for them mostly work on echinoderms as well. So they're not a problem. So where all the diversity really is in the ocean is in arthropods and mollusks. So there are about 70,000 marine arthropods that have been described so far and almost 2.5 times as many we expect that haven't been described. That's as compared to about half as many fish in the oceans. So all the crustaceans, and there are a small number of marine spiders, marine scorpions as well, so pycnogonids, account for a large proportion of the biodiversity of the ocean. Many of these species have very high reproductive output, very mobile larval stages, the sort of life cycle characteristics that you would expect from an organism that might be an invasive. So arthropods are a real problem. A classic example is the European green crab that is spread all around the world through shipping and having its larvae and eggs distributed as a result of that. Mollusks are a very similar problem. Mollusks are actually the largest, in species terms, the biggest phylum in the ocean. There are slightly more mollusk species, we think, than arthropods. Once again, many species have very high reproductive output and similarly mobile larval stages in early maturity. There are very long-lived ones as well, but many of them have these sort of characteristics. A classic IMS example is Pacific oysters. So these are spreading to Southeast Asia at the moment, and of course we're worried about them spreading to Western Australia. So one of the things that we did in this study was to look at the distribution of some of these species in Southeast Asia in ports that are adjacent to Western Australian ports. So this is a map of shipping with the density of the lines corresponding to how much shipping there is between different areas. And you can see somewhere, even like the Pilbara coast, which has very few people, has a huge amount of shipping going to and from Southeast Asia. So the density of those white lines is still pretty strong for that area. And that's, of course, because we're moving resources from the Pilbara to Southeast Asia and back again. And that creates a real opportunity for invasive species to be transported to this part of the coast. So we did some surveying of mollusks in the Pilbara as well as in Southeast Asia. So within the Pilbara, we didn't find many invasive species, which is great news, of course. It actually looks like the Pilbara coastline, as far as we can tell from some surveys that we've done so far, doesn't have a large number of invasive mollusks. We did identify new species of mollusks in that area, so a new species of this genus, which matches specimens in Queensland. Who knows whether that's a natural range or whether it has been introduced, but we assume it's a natural range. We also looked for mollusks in Southeast Asia that might become invasive in the Pilbara. So Magellana, which is the new genus name for Crassirostria gygas, the Pacific oyster. So it's been renamed, if any of you are used to that previous name. Sprat for that is farmed in Southeast Asia. So it's quite likely that that would be something that would become an invasive for the Pilbara, especially given that it occurs all up and down the east coast of Australia. So we did surveys there, didn't find it, but found close relatives of it, which is a great sort of positive control that going out and searching for it, you found other species that were native to that area, but we didn't find that invasive. Also found other IMS that were mollusks in that area, but not oysters. And in the upper Gulf of Thailand, the distribution of Magellana gygas was found to not be increasing through multiple years of surveying, which is great. So that's all very useful stuff. Another thing we did was to develop new technologies for identifying invasive marine species. So one of the classic ways of doing this is to put these settlement arrays into port areas. So this is this structure on the left with floats at the top, plates at the bottom on the quadrilateral structure where things can grow over time. You put these in the water, the things settle on them, grow, you scrape them off, and you can either look at them under a microscope or get DNA from them and examine that if you want. But these are really cumbersome things. It takes a lot of effort to do that. If you're looking at all of the stuff that you get off a settlement plate, if you're going to be doing eDNA analysis on that, there's quite a bulk of material for that. So Cheryl Lee did some work on developing these tiny little attachments, which one is on the settlement array for direct comparison, that you can allow the same kind of process to happen with. You leave them out in a port area for some time, things settle on it, eDNA accumulates in it. You can put it in the post it back to a lab like Shane's lab, Frontiers, and get some results from that and identify invasive marine species. So this is just a really great high throughput way of getting more information about invasive marine species of any sort. And that works really well. It works as well as the settlement plates as far as we can tell. We also developed some better eDNA tests based on some of the extra species reference data that Cheryl Lee developed from some of that sampling in the Pilbara and Southeast Asia. So some of the problems that we have with detecting things like crab species is that some of the classic primer sets that we use, like the classic 18S primer sets that is used for some of these groups around the world, is that you can detect that there are genera or families of these groups present, but you don't know what species they are. So basically the DNA marker doesn't have enough resolution to get you species level stuff. If you use some of the markers that do have species level resolution, it misses quite a few of the invasive species. So the primer set that Cheryl Lee came up with identifies more species basically, and it's allowed us to analyze more areas and potentially detect more invasive species, which is fantastic. We did apply this in a very large survey we did at Port Hedland. So we sampled the Port Hedland area, both in the industrial operations area, which is at the centre of this image, and in the riverine areas and some of the coastal areas just adjacent to that. We took over 1,000 marine samples in this area. We screened them all with this new test, and we found no invasive species, which is really great news. Excuse me. So just to summarize this first part of the talk, the impact of what we've done with invasive marine species is that we've developed better methods for detecting them, both better physical methods, better information to allow us to identify them when we do get them, better molecular methods for identifying the DNA from them when we can isolate that. We've built a network of researchers within Australia in our partner countries of Thailand, Singapore, Ascension Island, Fiji, Denmark, and Austria as a result of getting reference samples for many of these things. We've had some social and environmental impact by basically confirming that what you tend to think of as a fairly dirty port ecosystem is actually ecologically pretty good. It doesn't have many invasive species, possibly we hope, because it's sort of a bit buffered by having a healthy environment just adjacent to it, a really good natural environment in Port Hedland and the areas around there. And this leads on to plenty of things we can do in future work. So we're working on more better lab-based methods for invasive marine species detections. We're hoping to come up with a broad-scale panel that will detect anything on the world list of invasive marine species. And we're developing point-of-need detection methods for IMS species in the future so that you can actually go and do these detections in the field without needing to send things back to the lab. And there are certainly plenty of use cases you can think of where that would be valuable. So I'm going to hand over to my colleague Euan Harvey now, who will do the second half of this talk. And if you have any questions, we can both answer them at the end. Thank you.
First of all, I'd like to acknowledge my colleagues who have helped me with this, and particularly Tim Cooper, who put us on to the Pilbara Port Authority. So we've had a lot of help from Mark Logue and colleagues up there with the collection of samples and actually figuring out what design we should be doing. They helped us do a bit of experimental design. Simon has showed this. What we were trying to do was sample man-made structures, which in Port Hedland are the pylons and the rock walls, which have been made by the ports, and compare and contrast that with the natural habitats. So we've done mangroves up inside the harbour. We've done intertidal platforms just outside the harbour and up and down the coast. And then we've come over to this other system with site 14, 15, and 16, and sampled in those mangroves as well as another contrast. We sampled water, and we used rollers to sample the physical substrate. We ended up doing something like 972 samples split across both, so it's a really high intensity sampling program. And I'm going to mention the experimental design just a little bit because I'm going to come back to this. At each of our sites, we had 3 transects. Within each transect, we sampled an area that was at the bottom of it, at the middle, and as high up as we could go. And then within that, we did 3 water samples and 3 roller samples. I'm going to go straight into the results very quickly. With the 18S, we ran 3 assays, 18S, 16S fish, and then the one that Simon was talking about, I'm going to present data on too. There was no difference in the number of OTUs with the 18S, but there was a real trend towards the pylons and the intertidal reef having a much higher number of OTUs. With the fish, it was much clearer. There was a significant difference with the pylons being really different, the intertidal reef being different, and the others being very similar. What about an assemblage level? Well, again, with the 18S, which is at a broad phylum level, there were differences with habitat and depth, and the differences were between the intertidal reef in particular, which was really different, the harbour mangroves, which were further up. The others were similar. They were still statistically different, but what you also notice is the letters here are bottom, mid, and upper. So you start seeing that little bit of separation that's coming out with depth. It's not as great as the effect of habitat. Same story with the fish, only really a difference in habitat, and that sort of makes sense because fish are fairly mobile, they move around. I also think it might be an artifact of the sampling techniques, which I'll talk about in a minute. I'm just going to put this up. This is really hard to actually already get your head around. because there's so much data. But the red things, there's a lot of this sort of red. These tend to be organisms that are floating in the water. There's lots of different things there. We can't get the resolution to really understand what's going on. But as you move from the pylons, where you've got ascidians, sponges, anthozoa, polychaetes, and you start going across, you start seeing the complexity coming out, particularly in the mangroves, the intertidal reefs, and the mangroves that are outside of the harbour. We're going to explore this more, but not here today because we don't have enough time to do that. The fish was actually very similar. Again, we're going down to a family level where we can, and there was that difference between habitats. This actually becomes a little bit more easy to understand when you start looking at methods. The big thing that comes out is you've got all these well, basically what are sardines, pilchards. We don't know what types. There's lots of different types that are up there that could be any one of 15 or 20 different things because we don't have the resolution to get down to the level of species yet. I wanted to focus in a little bit on the differences between the water sampling and the rollers. And this is where we really do start to see differences. You'll see when we're looking, for example, just at the water, we're sampling lots of arthropods. We're also getting quite a bit of chlorophyta, chordata, annelida, mollusca, porifera. When you go across to the rollers, which we're rolling up and down part of a habitat like that, we're getting a lot more detail. And that sort of makes sense because the water's collecting things over a larger area, things are floating around. The roller's collecting things that are directly there. And when you start looking at the fish, there are things that start coming out that actually make a lot of sense. We're getting less of this, a lot more of other. We're getting down to a much lower level of species detection than the other, so that's a 2% cutoff. The thing that really comes out was when you look at the 18S and you start looking at the number of samples that are required, we've taken 972 samples. We actually needed more. So this is the water, and we're seeing we're getting up to a certain point. Rollers actually sample a lot more species. Combined, they sample more species again. So one of the key messages coming through is there is no one right way of sampling. You've really got to think about your question. In this case, we're doing broad diversity, and that's my justification for lumping the rollers and the water together just as a way of sampling, because we wanted to sample as much diversity as we could. And if we added another technique, we'd improve that diversity. So I think you really need to be aware of understanding what question you're trying to address before you choose a sampling technique. If I wanted to know about benthic organisms, I would go with the rollers. I'm coming back to this again. By taking the large number of replicates that we did, and you can see in those accumulation plots, there is a lot of variance between one sample and another. You'd expect that. How do you overcome that statistically? It's a mess to analyse statistically, and it makes it hard to detect changes over space or time. Within each of these areas, we had those replicate samples. So we ended up with 9 water across that bottom level, 9 rollers. And if we wanted, we could actually combine that with water and rollers, and suddenly we've got 18 samples. We've got 3 different platforms. Thank you. We've got 3 different platforms. What it means is we can move away from a sample being a replicate and into a site being a replicate and start getting frequency of occurrence. This is much more statistically powerful than 1s and 0s. And it's actually easier, more simplistic to analyse. We can turn that frequency of occurrence into sort of I won't say it's a rank abundance, but it's a rank frequency of occurrence and figure out what things are really contributing to the differences. So for example, if we look at the harbour mangroves, 56%, in 56% of the samples, we detected Sardinella, and then we detected Sardinella again. So one of the challenges with eDNA is we don't have the sensitivity to get down to a species level all the time, which we actually need with confidence. So I'm really excited that Ollie Berry and his team with the Future Science platform are actually doing something with the 16S fish. And I've given them our data, Simon's given them our data that I'm hoping to get back, and we can actually see what difference it makes, but it sounds like it makes a huge difference, which will suddenly mean that frequency of abundance could mean that we can actually set up a monitoring program where we can detect change. So what have we done? We've demonstrated that the man-made structures in Port Hedland actually have a really high biodiversity. There's no invasives. They have a unique habitat, which is similar to the platforms, similar to the intertidal reefs in some respect, but they're a unique environment. We've got a really robust baseline which can be used by people for experimental design going forward. And where do we need to go after this? Well, we've got to go beyond absence-presence into something that we can actually use to detect changes in the system and understand where those changes are occurring. So we need to be able to look at the development of a suite of metrics which can be used to describe that change. And it's not just absence-presence. There's so much more that we can do, and I know lots of people are looking at this around the world. If you want to know more, talk to Mike Bunce over a beer later, because he lives and breathes this. And with that, are there any questions?
Thank you. Is there any questions for Mike and Simon?
No, you can ask a question later over the beer. He can.
He can.
So I guess the main message from Simon's was that nobody wants to settle in Port Hedland. I have a question, I guess, for both of you, just around, like, both talks were around invasive species. I guess Euan's was more around also the general species assemblage. But do you have any thoughts on the detections, inferring where the detection comes from? Because obviously with invasive species, if you're in a port trying to detect, invasives in a port, and you detect something, it could be from a ship that's come in that has an invasive on it. The DNA is drifting from that invasive species into the water column in the port, and either being detected in the water or detected on the structures. So just interested in your thoughts on that, and also the passive sampler that you showed. Is the idea of that just as an early warning system? Because then you'd have to go and verify whether that detection came from a vessel, came from the port structure, or was in the water.
Okay, passive sampler, something that came out of New Zealand with WildLab, and it's an early warning detection. It's just trying to reduce the weight and the cost so you can put more out and get more early warnings. And Justin McDonald and Claire and Matt and others have been developing that over the last 10 years. So it's just an add-on. Point of source is a really hard question, and I would say that with water, I would be worried. It could be coming in from anywhere. We're not detecting those depth changes with water. With rollers, I think it's going to be very localised because we're going to detect those changes. So it's then a matter of setting up your sampling design in such a way that you're sampling the areas that are most going to be affected. Justin, if he was here, would turn around and say, well, it's the shallow areas because that's where invasive marine species tend to settle. So I would actually be using rollers over water for doing that sort of work, which is why the recruitment plates actually work well as an early warning indicator.
Just to add to that, if you do have a point of need detection system, then if you can get results back within an hour or so, you can actually do exploratory testing. So you could test, if you find negatives, then you can move on to another ship, another substrate, and search for detections elsewhere. So it's one of the arguments for developing point of need detection systems, that you can actually narrow in on a source.
Right.
Okay, thank you. Yeah, it's true, we do need to move on. So these guys will absolutely be at drinks afterwards, I'm sure. So, right, moving on. Next speaker is Dr. Mika van der Heijde and Associate Professor Paul Nevil. So, where am I? Next one. They'll be talking on the terrestrial ecosystem biomonitoring with eDNA across the Tree of Life, the Olympic Dam study site. So,
great, thanks, Shane. Moving back onto land now. So this project's focused about developing large-scale terrestrial ecosystem eDNA-based monitoring and Mika and I are co-leading the project, and we'd also like to send a big thank you to the site people at Olympic Dam, especially Kim Solly. She's been extremely helpful. And also, we left off Francesca Martino, who was a DREAM honours student that we had this year, and you'll see some of her work in a sec. So eDNA studies have been overwhelmingly in aquatic systems, either freshwater or marine. And one of the objectives of our group over the last few years is to bring eDNA-based monitoring onto land. So terrestrial eDNA studies come with their unique challenges. What to sample? There's a huge diversity of substrates that we can look at. Traditional ones are soil, water in lakes or dams, but we've also got new ones like air, and also vegetation swabbing is something that we've been spending a lot of time doing. Then also, where do you sample? eDNA in a terrestrial environment is distributed fairly patchily for a lot of those substrates. Where do we sample? Do we sample where the animals are if we want to look at terrestrial vertebrates, or do we sample quite broadly? And then how do we sample? Developing all the kit that Simon and Euan showed us, some of these new approaches like using rollers. Bunnings has been our friend throughout these projects. We're always hitting up hardware stores and making new things to sample eDNA. Then arid ecosystems present their own additional challenges. They're often really remote, so how do you store samples? How do you get sampling equipment out there? Low animal densities. If you're looking at terrestrial vertebrates, there's not a lot of DNA in the environment, and it's very patchily distributed. And also high temperatures and UV tend to destroy the DNA. So the aims of this project, which has only been running since January 2024. So this is quite a short one. We haven't had a lot of time on it. And also Mika's been part-time on it as the postdoc. So the aims of the project were to optimise eDNA sample collection from remote areas and large spatial scales, explore the spatial resolution of biodiversity data from different eDNA substrates, and also start working towards a terrestrial ecosystem health index. Social licence to operate is increasingly difficult to hold on for resources companies, perhaps not with Generalissimo Trump now in and Musk. That might change somewhat, but there's increasing emphasis on businesses looking at their impact on biodiversity and also the impact of biodiversity on their business, and the need for developing easily digestible metrics like the one at the bottom, which will show you how your site is tracking biodiversity-wise.
Okay, it's working. Okay, excellent. So I'll be going over some of the small tests we've done to try to optimize our research methods and how to sample and what to sample. So this was a small test we did because we've done some research with rollers in trees and in tree hollows. And we wanted to compare them to swabs to see, is there a difference? Does it matter if we use the rollers versus the swabs? They have some advantages because differently from air sampling, you don't have to leave something out for a long time. Okay, thank you. You don't have to leave something out for a long time and then revisit it after a week to pick it up. You don't need a powered vacuum or suction thing to pull anything through air. And you're basically collecting anything that's been deposited from the air onto these surfaces, leaves, trunks of trees, rocks if you want. And if you put them in a storage buffer, you don't need cold storage. So it's minimal equipment and you can detect a lot of things, including vertebrates. We found that there wasn't too much of a difference between the swabs and the rollers, which is pretty useful, although the rollers did detect more overall cumulatively, possibly because they have a greater surface area and you can have a greater reach. So if you put them on a pole as well, you can go all the way up the tree if you need to. The swabs, on the other hand, had the advantage of being smaller, already sterile, so you don't have to sterilize them and you can buy them in bulk, whereas the rollers, you have to pre-order them because each Bunnings only has about 10, and you have to go to 10 Bunnings to get enough for your project. The swabs are also cheaper because they also require less of the preservation buffer. So if you're going to pick we depend on what your limitations are. We went with the rollers because we valued the greater surface area and the greater accumulative diversity detected. We also did, this is the work of an honor student, Francesca Martino, and she was looking at air DNA sampling. And if you could just sample it from the car, you don't have to set up a pump or anything, just sample it from the car and go across a certain distance. She compared the car sampling with the stationary passive sampling. Can I do the, yes. No, it doesn't show up. The stationary passive sampling that we left either out for the same amount of time as the car was going or for a week for more long-term passive sampling. And she did it for 4 transects in the bushland and 4 transects in the agriculture, just to do that comparison of the different land uses. She actually managed to detect quite a lot of vertebrates, 49 taxa in total, including mostly birds, but also some mammals and 1 amphibian. Most of the taxa were detected in the long-term passive air sampling, so that was the better overall one, but a good number were detected from the vehicle-mounted sampling units and from the short-term passive samplers. There wasn't really much of a difference between the methods, richness per sample. So no matter which method you use, the car or the fixed passive sample, there wasn't that much of a difference. And this shows a lot of promise because if you're trying to sample a really large area, it helps if you don't have to visit the same area twice to set up and pick up your traps or equipment. It also helps if you can just drive a distance and switch out your mobile air collectors as you go, then you can get a lot of distance surveyed in a relatively quick amount of time, and it's quite efficient. So one of the other things that we were looking at was soil sampling. And so generally with soil samples, you collect a few subsamples, and you put them together, and you homogenize it really well, put it in another tube, and put that tube on ice. Unfortunately, if we don't have ice, or you're in a really remote location, that can be challenging. So we're trying to look at a few different preservation buffers. I'm mixing it really well. The problem with soil is that it's really heterogeneous, it's really patchy. So the reason you do a lot of subsamples is to try to get a general idea of the soil in that area rather than a soil of just that one specific point next to that one specific plant. That's why doing homogenization of samples is really important. And I am starting to see a little bit of that coming up in some of the water sampling when they're trying to do large bodies of water and they do a lot of samples and subsamples. So we were trying to test a bunch of different preservation buffers. And so far, all of the different methods, the cold storage and the 2 different preservation buffers that we're using, a homemade one that's slightly cheaper and a Kyogen one that we bought, all of them we could amplify bacteria, plants, animals, and some invertebrates and fungi. But that's still being processed, so I don't have all the community data from that yet. Finally, the project that we're doing that's still ongoing is a spatial resolution study. So this is at Roxby Downs, and we're comparing in the town versus in the bushland. And we had a grid of samples where we're looking at soil, passive air sampling, and vegetation swabs using the rollers. Basically, we have 180 samples because there's 60 of each different sampling type, and a second site that we're sampling in WA to look at the consistency of the different sampling types. What we want to know is which of the sampling types can tell us the difference between the different land uses, how far apart do the samples need to be, and how far from the adjacent land use do they need to be to reflect their current land use, or the land use that we are sampling at that moment. And this is the kind of information that we need in order to apply a more wide-scale sampling of different land uses to see how we can reflect the ecosystem conditions.
Thanks, Mika. So yeah, impact for the project, which has only been running for 6 months, is develop sampling methods that are fast, easy, reliable and cheap. We want to develop a sampling show bag that we can send out to a mine site and an enviro on the mine site who, didn't pass genetics at uni perhaps can actually go out and sample and get good biodiversity data from it. We don't want anything that's too complicated or just requires way too much equipment because it's just too hard to get it out in the bush. Minimisation, cold storage, also really important for us. And also information on spatial resolution of eDNA data. We want to know that this biodiversity signal that we're getting is actually local and not floating around from somewhere else. So all of this is really crucial for upscaling terrestrial studies. Future perspectives, we need to test this at larger spatial scales. We need to calibrate with other measures of ecosystem condition, whether it's remote sensing data or perhaps other methods. Multiple years of data consistency, the consistency of the signal in locations like Olympic Dam, water, rainfall drives everything. Whether you get a consistent signal over multiple years will be interesting to see. And then combining data from multiple substrates. Do we have a soil index of biodiversity? Do we have a vertebrate index of biodiversity? Do we have a plant one? Is there some way we combine all that data together into one? And that's it. Thank you.
Excellent. Thanks, Paul and Mika. I'll throw to the field if anyone has any questions.
Quick one. Thanks, guys. Amazing work. I was wondering, when it comes to eDNA, has there been any studies on the impact of elevation? So how far from ground level should the samples be taken? In other words, can you use a drone to collect samples?
I think there was a drone-based
Yeah, there has been
one
a drone-based study.
at some point. And there was an eDNA workshop in Canberra earlier this year that we attended. And someone did do a bit of vertical profiling, I think. And the message was the higher you go, the greater the area that you're sampling because it's coming from further afield. And the lower you go, the more local it is for the most part.
So if you want to get an overall, like a landscape idea, you could use a drone with the same filter, the filter system that you drew around with?
Yeah.
Theoretically?
Theoretically, yes, but you would also have to have that drone flying around for a while.
Of course, yeah.
And that would probably drain the battery quite quickly.
Yeah, true.
The cars last a bit longer compared to drones, is my understanding.
Fair call.
But it can't go as high, yes.
Thanks, that was a really interesting talk and tool. Is there any plan to look at the inverts, like the insects specifically with the eDNA samplers? It's like you did for soil, I think you did a cross taxa, but I think you just presented results for reptiles, birds and mammals from the air.
Yes, so the swab test, I just did the 18S because it's a really broad eukaryote assay, and I did the 12S because people are most interested in birds, and if you want people to be interested, you do birds. The other one with the car thing was also vertebrate focused, partially because it's an honor student program, and they're most interested in vertebrates. We are planning to do as much as we can with the invertebrates. The challenge with invertebrates, especially on land, is the reference databases, right? Most of the invertebrates don't even have names, let alone sequences. So we'll get a lot of sequences, but we won't be able to assign them. And most of the assays that detect the inverts also pick up fungi and a few other things. So you can't say that this assay shows all of these potential inverts. It could be 30% are inverts and 70% are fungi. So that's where the challenge is with inverts. We need to develop those reference databases more.
Thanks. And we're working at scale, just a plug for Josh Newton's PhD. We're driving a transect on Sunday to the South Australian border and changing out samplers every 20 kilometres. So we get a good picture of biodiversity, hopefully across the whole 1,300 kilometre transect, and then we're going to sequence vertebrates, plant DNA, fungal DNA, etc.
Thanks, guys. Ollie Berry here. Terrestrial eDNA is definitely the final frontier, and it's more a suggestion than a question. I think I've got a great sponsorship idea for you. Why don't you team up with Australia Post or DHL, affix some sort of, like device on the windscreen? Because what I'm thinking about is these fixed transects at these companies are driving day in, day out. You know, it's an opportunity to monitor the nation.
Yeah, we love that idea. We've been thinking about that for a while, getting trucking companies to put samplers on. But yeah, Josh's project will be a proof of concept on how well it works. And yeah, good idea.
Excellent. All right, thank you, Paul and Mika. All right, the next speaker will be Dr. Angus Lawrie, who's talking on, I know you're not supposed to pick your favourite kid, but I think this is the latest kid in the portfolio. So this is a really exciting project on the living waters of Western Australia.
All right, good afternoon, everyone. So I just want to see what's going on. Yeah, sweet. Oh, anyway, missed some title slide. No, come back. So yeah, I'm the project manager of this project, but there's a lot of other people that have contributed to this work. This work has only started within the last since the first half of this year, so I don't have many results to present today, but this is more about a forward-looking, what we're trying to do with this project and what it is. So Living Waters West Australia, I'm going to refer to it as LIWA because abbreviations are fun. So we're working on wetlands, and basically we're actually trying to solve some of the problems that we've talked about today already. So we're trying to develop an eDNA monitoring tool for our wetlands, but we're going to do it in an interesting way that I'll get to in a minute. This project has a few different components. The second component will be run by Dr. Matt Campbell, who will be looking into how biodiversity has changed in our wetlands over time, using ancient DNA from our sedimentary cores, both looking in the recent past and then hopefully exploring those changes over a much larger time frame. And then a really interesting component of this work that I'm really passionate about is spreading the good word of how interesting and amazing the biodiversity of our wetlands are. So the last component of this is a community engagement program where we're going to try and engage with as many different stakeholders as possible. Today, I'm basically going to be focusing on this first application, though, this idea of this monitoring tool. So why wetlands? Well, historically, wetlands have been great places for us to fill in, build houses on, and turn into farms. Here in Perth, as an example, we've destroyed 70% of the wetlands that were here prior to colonisation. And even the ones that are left, a lot of them have been used as landfill sites or as areas of drainage for our runoff. So we've done quite considerable damage to these ecosystems. What we also know about these ecosystems is that they tend to be quite diverse and poorly studied. Those two things together make it very difficult to monitor changes in them effectively. So I guess the problem is, well, what's eDNA got to do with this? eDNA is fantastic, right? We love eDNA. It's the best. It's got great applications in biomonitoring. And that is all absolutely true. But as we heard before, there can be consistent problems with trying to understand when we sequence something in an eDNA context, we're relying on somebody else who's already looked at that thing and put it into this wonderful reference database so we know what it is. So often when we sequence material from the environment, we're only getting back a small proportion of the actual sequences that we've actually generated. That doesn't make sense. But what I'm getting at here is, this is an example. So this chunk up here is the chunk of sequences that we have been able to put names on for this preliminary work that we've done so far in our wetlands. But this is the proportion of sequences that we actually have. So the question is, what information is being lost in all of that sequence data that we don't know what it is, but it's all real DNA and it's all in those ecosystems. So what can we actually find out that's in that information? And this is kind of the crux of this index that we're trying to work with. So can we use that lost information? Before I get there, I just want to give a very quick indication of what generally biomonitoring indexes are usually composed of. So just as an example, we might have, if we wanted to develop an index of wetland health, we would go to wetlands of lots of different types. And we would see, for example, that some wetlands have, might be in a nature reserve, be full of fish and frogs, maybe not in the Australian example, but typically speaking. And then we might go to some other wetlands that might only have kind of worms or more hardy taxa that will live kind of anywhere. And basically what we'll do is we'll go to those two different communities and we'll characterise the animals that are there. We'll say, When it has fish, it's good. When it has worms, it's bad, very simply. And we can put a number on the occurrence of each of those animals. And that's great, and that works, and we've been doing that for a long time. The tricky thing with this is that having the expertise to go out and do that work is you actually need to know what you're doing. It's quite expensive, and all the other usual things that eDNA people will say about traditional methods. But there's this emerging really interesting idea that that fish is in this lake, but so is its DNA, right? And that worm is in this lake, and so is its DNA. So I can still say, if I detect that fish's DNA, it still indicates that this wetland is in good health, right? I can say the same thing for the worm. I can say the same thing for the frog. I can say the same thing for the snail. But then I can also make the same association for all the stuff that I don't currently know what it is. It's very simple. If the thing that I don't know what it is, and I don't need to know what it is, only occurs in good lakes, then that DNA sequence still indicates good health, if that makes sense. So that's the basic premise of what we're trying to do here. We're trying to move away from the idea that we necessarily have to know what a DNA sequence corresponds to, and only need to understand its association with particular habitats. This is what we generally call a taxon-independent index. Some very clever people in New Zealand came up with a really good version of this index for New Zealand rivers, and we're basically trying to see whether or not we can apply this same premise to Western Australian wetlands. So as I said, I don't have any results really to show you. I have some preliminary data that I'll get to in a minute. But basically what we've done so far is we're trying to generate this model, this index, for wetlands in Perth, in the greater Perth region, for about spring and winter time. So what we've done so far over the last field season is go out into 65 lakes around the greater Perth area, collect lots of eDNA samples, and these lakes have been, disgusting cesspits to some beautiful nature reserves and everything in between, and that's quite on purpose because you need the good to understand what the bad is and vice versa. So what we've been doing at each of these lakes is we've been taking, as well as our water samples for eDNA, we've been doing macroinvertebrates, and then some more classical water health kind of parameters to build a more comprehensive picture about what type of wetlands we're sampling so that we can calibrate our eDNA results against more traditional ways of understanding what wetland health is. One of the really interesting parts about this study is that we're not just looking at microbes or fish or insects, but we're trying to do a quite tree of life analysis. So there's 14 different assays that are going to amplify everything from microbes to fish to insects to everything you could possibly imagine. And that's a lot of data. And basically the idea is we're going to feed all of that massive amount of information into our model that's going to look at the community assemblages, and then we're going to be able to come up with an idea of what lakes are healthy and what lakes are not healthy. That's the theory. Just as an example of how things are going so far, so these are just two sites as an example of what we've been doing. So Lake Monga, just around the corner from here, has a standing algal bloom, has been a historical landfill site, is truly disgusting. Lake Quenda Swamp is a much nicer wetland that's in remnant vegetation. It's relatively undisturbed in comparison to a lot of other sites. And so our prior would be that Quenda Swamp should be more healthy than Lake Monga. And indeed, when we chuck all of our data into our model, we get a situation that suggests generally that same trend. So it's hard, the absolute numbers don't necessarily matter so much, but the point here is that the number on here is lower than that number over there, which tracks with our prior as to what we think these environments are relative to each other, if that makes sense. So far it's working well, and we're really interested to see what happens when we go to all of the data back for the chunk of sampling that we've just done. And then just very quickly, I'm not going to go into this just yet because it hasn't actually started yet, but Dr. Matt Campbell will be running this project where we're going to be interested in looking at biodiversity changes in our wetlands over time. So we're thinking of two different timescales, one since European colonisation, so over a shorter time frame, and then seeing if we can go back much further to understand how biodiversity has changed in these systems. And my favourite part is I get to go and tell people how great these wetlands are. And so that's going to be a really key component of this study. We're starting this on the 22nd of November in the Wetland Centre in Coburn, if anyone's interested. And on a personal level, I've already taken my dad out to do a lot of this work, and if a social scientist can embrace it, then I reckon anybody can. So thank you very much.
Excellent. Thank you very much, Angus. Did anyone have any questions for Angus? How are we going online, incidentally? No questions. No. Ollie. For the online people.
Thank you. It was a great talk. And so the tiki in New Zealand is super impressive and inspirational. And I'm just wondering, so the Swan Coastal Plain is quite stratified. It's got these micro biogeography, if you like, with these dune assemblages. And I'm just wondering if that's going to add a level of challenge to how you assign what's healthy and what's not healthy, because they may be multiple versions of healthy depending whether they're in spearwood dunes or basandine or whatever. So yeah.
That is the single greatest challenge of this project. How do you disentangle biogeographical differences from environmental differences? So the key assumption to this project, right, is that our differences in community composition will be due to some disturbance gradient. When you're dealing with rivers, I think you'll have a separate situation than when you're dealing with lakes. Lakes tend to be evolutionarily islands, so you get larger, small spatial scale differences between sites, not because they're necessarily that different in a disturbance factor, but just because they are inherently different, as you say. So this will present significant challenges. The way that we're trying to factor that in is to, as much as possible, stratify our sampling regime so that we have representatives of as many different ecosystem types or lake types as possible. So you can imagine you have a permanent lake will be different from a seasonal lake, just purely because they have different hydrologies. A saline lake, which I come from a salt lake background, I care a lot about salt lakes, but I've completely excluded this from this project so far, just because that adds an entirely new element. So what we're trying to do is wrap our hands around a lot of variation. And I think that the key thing here will be, are our sample sizes big enough to wrap our hands around that amount of variation that is inherent to these systems before we can even understand whether or not that variation is being driven by disturbance. So it's a good question, and I've got no idea.
That's nice. Were there any other questions?
Maybe I can comment, because some of the data you had already, Angus, is quite promising, I would say, where you actually see some sort of correlation that is expected, right? The ones you expect to be poor also show up with a low number in the tiki. And then you have outlier sites that doesn't fit the pattern. And then now we can kind of retrospectively go back and see why the hell do these not fit the pattern? What kind of biotic or non-biotic factors are probably forcing these guys to sit outside elsewhere in the correlation, right? So I think we will have to tackle it a little bit like that over these.
Yeah, no, I'm being a little bit facetious. We have seen that it's broadly doing what we expect it to do, but the outliers are interesting in the sense that, are they different because of environmental reasons or other reasons? And with a lot more data, which we're about to get back in a few months' time, I think we'll tackle a lot of those questions.
One other thing would be, what's the indicator on species number for this reference set?
Yeah, thanks for that. And I was involved in, I guess, formulating the tiki in New Zealand. And if you'd asked me a number of years ago whether we would have been able to take rivers from the bottom of the South Island all to the way to the top of the North Island and develop a single index off metabarcoding data, I would have said no, they're too diverse away from each other. But that is actually not the case. So the 3,000 indicator sequences that form the tiki, and typically you get between 200 and 500 indicator species per waterway that you sample. And about a quarter of them are bacteria, a quarter of them are microeukaryotes, a quarter of them are arthropods, and the other ones fill up the rest of the thing. There is enough diversity in those metrics to span a huge diversity of biomes that occur in there and still get a metric. And I guess what some people are missing a little bit with the tiki is that it's a triage tool, right? It's a flag that you put up and say something's going on here or it's changing, and then you might go in and do a more intra-lake variation, right? But without that flag that gets raised up, you just don't ever go there. And that's the bigger crime there, that we miss changes in condition without raising flags. Thanks.
Great. Thank you. All right, thanks Angus, and thank you to all our speakers. So that concludes the scientific presentations for now, so we might as well give everyone a round of applause. So now I'd just like to introduce Dr. Tim Cooper, who's the Head of Biodiversity and Land in the Environment Group at BHP, to say a few words. Tim, it's hard to say how much we appreciate his help. He's been absolutely instrumental in this initiative and continues to be a really good guidance in each of these projects to the university's team. As I understand it, he's also been busy, I'd use the word bunts, but not everyone's going to get that in the audience, but he's been busy spreading the good word of eDNA enthusiasm all around the world through all your various channels. Which is, with the more eDNA-based tools like the ones that you've seen here today, biodiversity is going to be all the much better for it. So without saying much further, I'll hand over to you, Tim. Thank you.
Thanks very much, Shane. Shall I just stand here?
Yep.
Is that okay? And this works by that one. Okay. Yeah, look, thanks very much, Shane and team for the presentations. We had a call on Friday and was that, are we all good to go? And the team had a really great idea. They said, we want you to sort of summarise and describe the impact of this project. And I thought, wow, how do I go about doing that? So I had some notes and a speech prepared, but I've just realised that our ESG legal counsel is in the room. So I'm just going to... I didn't have time to get my speech approved, so I'm just going to step away from my notes and just share some random thoughts with you. So yeah, look, first of all, as Morten mentioned at the outset, this is a project funded under our social investment framework, the strategy, and it's informed by our strategy. When this project kicked off, we had 3 pillars in the strategy: environment, community, and workforce. And as a former academic, as Shane alluded to, it would have been negligent of me not to try and grab as much of that environment budget and channel it into science. And that's what we did with this project. So 4 years ago, we kicked it off and we had a lot of ideas, and thank you, Morten, for reminding us that COVID happened in the middle of this project. So in terms of impact, and look, we really look for projects that generate impact, and this one kicks so many goals and is really the gold standard of social investment projects in BHP, in my mind. So as it says up there, there's been 22 scientific publications. The project supported 6 PhDs and postdocs. There are 10 research staff that have been supported. I love the sequencing of the Pilbara Olive Python genome. World first, I'm assuming, yes. 391 marine sequences made available in GenBank. Over 1,000 samples, Euan, taken in Port Hedland. I think that's just amazing, sort of where that project started. We had project, I think it was 5, and we weren't quite sure what to do with it, and we sat down and said, let's just go sample Port Hedland, man-made infrastructure, structures and natural environments. And of course, Nicole, your great work underground. Just fascinating to see how excited you get over isopods. and that dot on the screen, amazing that you do that work. Yeah. The team have been to 12 conferences and workshops, and we've done this meeting 4 times. So with no disrespect to my team, the broader team that I'm in, my supervisor, who I hope is on the line, this is my favourite meeting of the year. The single best event I come to. I love it over and above everything else that we do at BHP in the environment team. So just Sam and Morten threw together some photos here. I love the Danish Steve Irwin over there wrangling that snake. That's one of my favourites. I've used that internally. This concept of putting filters on cars, I mean, we've got a big train network. We can put them on trains as well. So, just throwing that one out there. I love the marine environment. Most of you know that, well, most of you might know that I come from a marine background, and this is actually where eDGES started. I had this idea about getting better at understanding invasive marine species. And one of our previous leaders, who's no longer with BHP, she retired last year, said, great idea, Tim, but why don't you make it bigger? So go and get more ideas, and that's sort of where eDGES as a concept of projects came from. So thank you, Anne, if you're listening in. Yeah, the bubblers, they're going and sampling in the Chilean wetlands, and these photos of sampling in salt lakes this has been a really great project, and I have lived my life as a former academic vicariously through you guys, so thank you. And yeah, look, it does give me a whole lot of pleasure and excitement to announce that we're going again on the eDGES program. So about, what, was it 9 months ago, Euan and Shane came to me and said, look, this project's going to wrap up this year. What should we do? And I was like, well, let's just do it again. And so, but this time we're going bigger. So we've got 9 projects now, and I'll just flick through them in a minute. Supporting more staff over a slightly longer time frame, and about a $7.8 million budget, which I'm super excited about, to be able to put that into science in Australia, but also globally as well. So what are the projects? So project 1 will still have a reptile monitoring component in the Pilbara. Just excuse me for a sec while I put my eyes on so I can read this. I had it in my notes, but I had to step away from my notes. So. Project 2 will continue the great work that Nicole has spoken about this afternoon on underground fauna detection and conservation. So much new species potential to be discovered there. I'm really excited by that. We continue the work in Chile on the Salada Punta Negra and really take a restoration approach to that. The IMS project continues, so project 4, taking that into new areas. I'm not sure, I arrived into the room late, so I'm not sure if Fred is here. Is Fred here today? He's in Melbourne. Okay, project 5 is going to scratch a major itch of mine I've had all my life, and that is writing a book. Obviously, I'm a bit too busy these days to write books, but Fred is going to take that and pull together something that I think he's wanted to do pretty much all of his academic life, and now that he's retired, and that is a marine life of the Pilbara book. So that's going to be super interesting. Project 6 continues on, so that's the work that Mika and Paul spoke about before around monitoring land ecosystems, that new frontier, as Ollie referred to. Angus, you're a hard act to follow. That was super entertaining, your talk. The Leeward project's just kicked off. Can't wait to see where that goes. That will have as he described, a monitoring of wetland health and developing an ecosystem health index component, as well as the ancient DNA. This project will also pull in components with traditional owners and community. So really excited about where that one goes. And we'll pull in as many BHP leaders into that project as we can. So when we get executives coming over, we get them into waders and out sampling eDNA. I'd like to get our CEO doing some water sampling on Project 7. Project 8 really pushes into that question that someone asked before about species abundance. And number 9 is a bit of a moonshot. I think we had this idea late in pulling this proposal together, but it's thinking about how can we use eDNA in natural capital accounting decision-making frameworks. So really excited by that and where that opportunity can take us. So Project 9 will sort of really talk about that crossover. I think, Morten, you referenced it earlier about sort of how do we integrate across other disciplines. So on your question at the start, has this been a major waste of money? Absolutely not. And yeah, look, I think that is it from me. So look, I saw this slide on Friday and I thought, it's a pretty boring slide. It's a long road, but I think it's a long but fun road ahead of us. And certainly the talks that we've seen today will hopefully take us along that journey. Morten, would you like to come up and say a few words?
Just give her a hand. Yeah, 4 more years. How exciting. Thanks so very much for announcing that, Tim. Of course, we've known this for a while, right? But this gives great pleasure that now we can finally announce it publicly that eDGES is not stopping this afternoon. We're actually continuing for another 4 or 5 years doing what we love. So we're very excited about that. Looking very much forward to the continued collaboration and keep going. Yeah, and as you mentioned, they're also implementing some of these other aspects that are so important. It's not only about producing hardcore research, we actually need to translate that into politics and into other types of outcomes, right? So this is what eDGES version 2 will be partly focused on. We still need that foundation of great hardcore primary science, but there will be other elements implemented here that we haven't really touched much upon in the first version here. I'm not going to talk more now. I think this is a wrap for me, and I just want to thank everybody again for showing up this afternoon. Thank you so much to all the speakers we've had today, and let's just give everybody another round of applause for that. And we definitely anticipate to keep having these workshops also in the future, so I hope to see many of you back around this time, probably around this time next year. And now it's beer time, right, Shane?
Absolutely, yes. So yes, we will be continuing these, so any feedback anyone has, then we'd be happy to talk about it in the next little while, but for now we'll call a wrap, and so we'll move down for those of you that are able to stay on. W. Churchill's, so we'll get some of the team to show you. Down the stairs, across a little bridge, and then it's there. So we look forward to seeing you there. We've got a section in W. Churchill's there. Thank you very much for coming. It's been the biggest crowd we've had yet, so it's fantastic to see all the enthusiasm. So please stay around. Thank you. Bye.